Hello everyone, this is Mukul, your botany mentor for upcoming 45 days. And today in this power hour session, we are going to study two main chapters. It is a basic chapter, but weightage is high. The first chapter is cell, the unit of life, and second chapter is cell cycle and cell division. I'm giving you a quick recap what we are going to discuss in today's session. First of all, we are going to study the discoveries of scientists, various scientists, their year of discovery and discoveries. Then in next slides, we are going to discuss some scientist background, what they are going to discover, and on the basis of that, I have some questions for you in this slide, as well as today, I'm going to discuss some NCERT key points. NCERT diagram, NCERT key points, as well as I have some handwritten notes for you. So this session is going to be bang session, and let's start with the first slide, in which I am going to introduce you what we are going to do in upcoming 8 days or is 45 days. First lecture 11th March 2025, I am going to cover first topic, that is the cell, the unit of life, and cell cycle, cell division. If we consider the marks or weightage, this cell, the unit of life, almost time in past year they are asking 3 to 4 MCQ. It means it counts 12 or 16 marks, and cell cycle, cell division, from past years they are asking 2 to 3 MCQs. So nearly 12 marks from this chapter. So today we are going to study the two chapter, and in first slide, in very first slide, I am going to discuss some 10 to 11 scientists of this chapter, years of this discovery, and what they discovered. First scientist, that is the Robert Hooke, and in 1665 he discovered dead cell. Very important point, dead cell. Understood the thing, dead cell, and where? In cork of stem. Second scientist, Anthony van Leeuwenhoek, in 1674, after the discovery of Robert Hooke, he discovered living cells. So if in NEET they ask you the question, who discovered dead cell? Your answer is Robert Hooke. And if they ask who discovered living cells specifically, then it is Anthony van Leeuwenhoek in 1674, and where it discovered free living alga, that is known as the Spirogyra. Free living alga, that is known as Spirogyra. Then, very crucial MCQ, Robert Brown, 1831, discovered the nucleus. Third important point. Fourth, M. Sheldon and Son. Sheldon and Son gives us a cell theory. You know that. M. Sheldon in 1838 told us that plants are composed of cell, and Sean in 1839 discovered the plasma membrane and cell wall. You know that Sean also said that cell wall is a unique feature. Cell wall is a unique feature of the plant cell. Then after, Rudolf Virchow in 1855 gives us terminology that the omnis cellula e cellula. It means every new cell formed from pre-existing parent cell. Omnis cellula e cellula. Then Fleming in 1879 discovered a chromatin by using the basic dye. Hans Christian Gram 1884 gives us a gram staining method to distinguish two types of bacteria. Which are those bacteria? Gram positive bacteria and gram negative bacteria. After that, Camillo Golgi, you know that Golgi complex or Golgi apparatus discovery in 1898. Then George Pallet for ribosome discovery in 1953. And last 11 number scientist is Singer and Nicholson in 1972 fluid mosaic model of the plasma membrane. So you understood all the scientists, their year of discovery, and discoveries from this topic. Now, specifically question asked on the 4 to 5 scientists. One is the Robert Hooke, discovered cell, or we can say that dead cell. If they are asking dead cell, your answer is Robert Hooke. If they are asking about cell, then also your answer is Robert Hooke. But specifically, if in NEET examination they ask about the living cell, then your answer is Anton van Leeuwenhoek. Then Sheldon on 1838 observed plants are composed of cell. And Sean observed that, or Sean gives the hypothesis that animal and plant are composed of cell and product of cell. And this is very, very crucial point, that cell wall is a unique feature of plant cell is given by Sean. Now, by using two studies, Rudolf Virchow on 1855 gives us the terminology that is omnis cellula e cellula, and he modified the cell theory by using two statements. Rudolf Virchow modified a cell theory, and what is the modification, or what are the statement which is given in a cell theory? First statement, that all living organisms are composed of cell and product of cell. And second statement, all cell arises from the pre-existing cell. It gives us the information about how cell are formed or how cell divide, but it is not given us the information about how first cell is discovered. So it is given in the chapter of evolution in the zoology section. Now, first MCQ in front of you, on the basis of data. You understood the data of the scientist. You have undergoing the data of this complete chart. Now question is front of you. Question is, match the column 1 with the column 2 and select correct option from the given table. Table is there, column 1, column 2, scientist name and discovery, and you have only 10 seconds to solve this MCQ. Go for it, guys. I am with you. I am in a chat. Yes, everyone, what is the answer? 21 students joined in the chat. You have time of 10 seconds. Okay, done. Let's solve this problem. First scientist, that is the Leeuwenhoek. You know that he discovered the living cell. Any statement given us the information about that? First saw and describe living cell. It means A3. Then second, Robert Brown. You know that he discovered the nucleus. Any statement? B3. Then Sheldon and Son. But on the basis of those two options, we are going to solve the MCQ. Leeuwenhoek 1, A1 in this 1, B and C, so not in C, in D option. Second, Robert Brown 3 means B3. B3 in those two options means we are on terms of A and B, confused in A and B. Now we are going towards the Sheldon. Sheldon. Presence of cell wall is a unique feature given by Sheldon or Son. It is given by Sheldon or Son. It is given by Sean. So D is 2 and Sheldon give all plants are composed of cell. So correct answer is A. Correct answer is A. Very nice. You are answering in chat. You are giving the answer in chats. Okay. Let's go for second question, guys. Let's go for the second question. Second question in front of you, statement based question. There are 5 statements and you have to choose the correct statement. There are 5 statements and you have to choose the correct statement. 10 seconds for this MCQ. Go for it. Everyone. I am in a chat. Go for it. Answer the question. 5 statements. 3 are correct, 2 are wrong. Yes. Some students in the chat section trying to answer the question. Can you answer this question, guys? Okay, let's solve it. First statement, the shape of cell may vary with the function they perform. Yes. On the basis of function, it means if nerve cell is responsible for conduction of impulses, so it is elongated cell, right? RBC is responsible for carrying of oxygen, so it is biconcave in shape. So yes, the shape of cell may vary with the function they perform, so it is a correct statement. Second, the prokaryotic cells lack all membrane bound organism. Definitely true. If we are talking about the prokaryotic cell, membrane bounded organelles is not present, so this statement is also correct. Now third statement, Sheldon, a German scientist or botanist, concluded based on his studies on the plant tissue that the presence of cell wall is a unique characteristic of plant cell. It is given by Sheldon or Son. It is given by Sheldon or Son. It is given by Sean. So it is a wrong statement. C is not a correct statement. Now coming towards the point number D, based on their studied on a plant and animal, Sean, a British zoologist, proposed the hypothesis that the bodies of animal and plant are composed of cells and products of cell. It is a true statement. Yes, it is a true statement. It is right. It is the NCERT line on page number 2, Cell the Unit of Life chapter. Last statement, A, B, D are correct, but last statement, Sheldon and Son modified the hypothesis of Rudolf Virchow and gave the cell theory a final shape. Is it true? Or it is reverse? Rudolf Virchow modified the hypothesis of Sheldon and Son? Yes, Rudolf Virchow modified the hypothesis of Sheldon and Son. So it is also a wrong statement. It means A, B and D are correct. Any option A, B, D? Yes, option number D is a correct answer. A, B, D is a correct answer. I hope so you understood the concept of scientist or scientific background on the basis of those two very important and crucial questions. Now coming to the next important point or next important topic, those are very crucial point in NEET examination. They are going to ask you the question on the basis of these points. First, smallest cell, mycoplasma, that is PPLO, pleuropneumonia like organism, and 0.3 micrometer in length, very small, smaller than bacteria also. Largest isolated single cell, egg of ostrich, very nice. Longest cell, just before some time I explained why nerve cell is longest, because it have to carry impulses. So longest cell is nerve cell. Size of bacteria, 3 to 5 micrometer, and human RBC largest 7.0 micrometer in diameter. So look at the data. On the basis of this data, I am going to ask you one very crucial and very important question. Very important and very crucial question. Let's discuss the question. Everyone on the board, very important question. Different cell have different size. Arrange the following cell in an ascending order. Ascending order means smallest to largest. Ascending order means smallest to largest. You have option in front of you. You have a 4 cells, mycoplasma, ostrich, human RBC and bacteria, and those are the option. 10 seconds for you. Everyone, 10 seconds. 21 student joined in this live session. 21 students are there. I'm expecting answer from you guys. I'm expecting answer from you. Which one is correct answer? Okay, number 1, Ruchi Jha ma'am, our chemistry faculty answer that answer is 3. Let's check answer is 3 or not. Samir Vlog answers that answer is A. Okay, fine. I'm expecting more student to answer in the comment section. Anyone? Go for it, guys. Some very important question I have in upcoming slides also. Assertion, reason questions, statement based questions. Match the pair, match the columns. Okay, let's discuss the answer. Let's discuss the answer. Egg of ostrich is the largest cell, so it comes at last. Smallest cell is mycoplasma or bacteria. I just told you mycoplasma is the smallest cell. Mycoplasma is smallest cell 1. Second is bacteria. Third one is a human RBC, and fourth one is a ostrich egg. So 1, 4, 3, 2. 1, 4, 3, 2. Any option? 1, 4, 3, 2. 1, 4, 3, 2. A is the correct answer. According to me, A is the correct answer. Let's move towards upcoming slide. Let's move towards upcoming slide. Now we are going to discuss a prokaryotic cell. So what do you know about the prokaryotic cell? they have no membrane bound nucleus and organelles. No membrane bound nucleus, no membrane bound organelles. Second, they include, first, almost all bacteria are prokaryotic in nature. Blue green algae, that is the cyanobacteria. Mycoplasma is also known as PPLO, pleuropneumonia like organism. Those all are included into the prokaryote. Then basic shape, bacillus, that is the rod shape. Coccus, that is the spherical shape. Vibrio, that is the comma shape. And spiralum, that is the spiral shape. So in that, sometime NEET is going to ask a question on the basis of coccus, that two coccus, two spherical shaped bacteria joined together. Then it is known as the diplococcus. If there is a chain of the coccus, then it is like a strip. So on the basis of that, we are called it as a streptococcus. And if there is a bunch of coccus, bunch of spherical shaped bacteria, then it is known as, in comment section, then it is known as Staphylococcus. Then it is known as Staphylococcus. Okay. Now next point, cell envelope is made up of three tightly bound layer. First one is the glycocalyx, second one is the cell wall, third one is the plasma membrane. If I am asking you the question, glycocalyx divided into two parts. One is a smooth, one is a tough. One is the smooth and second one is the tough. Smooth glycocalyx is known as slime layer, and the tough glycocalyx is known as capsule. Let's move forward. Hans Christian Gram, a scientist who discovered a method of gram staining. There are two types of bacteria. One is the gram positive bacteria. One is a gram positive bacteria. Second one is a gram negative bacteria. Second one is gram negative bacteria. So bacteria which retain gram stain is a gram positive bacteria, and bacteria which is not retain gram stain is a gram negative bacteria. So which kind of conclusion we are going to understood from this concept? When we take the antibody, if any gram positive bacteria affect us, it easily retain antibody and easily killed by the antibody. But if there is a gram negative bacteria, it does not retain the gram stain. Means it is not easily retain the antibody. And if it is not retain the antibiotic, it means it is hard to kill. It can be killed, but it is difficult to kill a gram negative bacteria. So strongest bacteria is gram negative bacteria. This is the conclusion. Now, a very important concept, that is a eukaryotic cell membrane. This concept, on the basis of this concept, questions are going to be asked in the NEET examination. And in 1972, Singer and Nicholson, two scientists, give us a model of plasma membrane, very known model of plasma membrane. Can anyone give the name of this plasma membrane given by Singer and Nicholson? Year is 1972. Singer and Nicholson gives us the model of cell membrane or plasma membrane. This model is known as, in comment section I am waiting for this. 17 students are joined in the chat. In comment section, I am waiting for the answer. Anyone? Anyone from class? Anyone from joint student? The answer is fluid mosaic model. The answer is fluid mosaic model. And Rugved gives us the answer, that is AMB, fluid mosaic model. Very good. Rugved, AMB, answer the question. Before that, Muskan, Sabha is also answering the question. Correct, Sabha, correct. Fluid music model. Thank you so much, Suraj. Thank you, Suraj. Aditi, fluid music model. Very good, student. Very good, very good, very good. Let's understood the fluid music model of the plasma membrane. What they are saying in the statement, first statement, chemical study on the RBC show that the cell membrane is composed of lipid bilayer, that is a phospholipid bilayer. Second one is the protein. Two type of protein, integral protein and peripheral protein. Third one is the carbohydrate and some part of the cholesterol also. Now, lipid mainly is a phosphoglycerate. That's why I told you phospholipid bilayer. Now, have outer polar head. Polar means hydrophilic in nature, water loving molecule, and non-polar, that is a hydrophobic tail. It means hydrophobic, water hating molecule. So let's draw here a plasma membrane. If I am drawing one circle and this is a phosphorus, this is the phosphorus head, so we can call it as a hydrophilic in nature, hydrophilic in nature. And if I am drawing here tail portion, then this tail portion lipid tail, and this tail portion we can call it as hydrophobic tail, hydrophobic tail. So hydrophilic head and hydrophobic tail combines together to form a fluid music model of the plasma membrane. Let's draw this model. If I am drawing here some portion of phospholipid head and some portion of hydrophobic tail on lower side, because it's not a single layer, it is a bilayer. Bilayer means on above side one layer, on lower side also one layer. So I am drawing the another layer, that is a bilayer. Here 1 2 3 4 5 phosphorus head and some part, that is the tail portion. So this is a phospholipid bilayer. Now we know that some integral and peripheral protein are also there. So let's draw one protein molecule in this diagram. Another, I am going to continue diagram. I am going to continue this diagram with the phospholipid bilayer. Let's draw one another integral protein here, and let's continue this diagram. With the hydrophilic head and hydrophobic tail. Let's assume that this is a fluid music model, or this is a plasma membrane, and now on that some protein are situated on this point, that is known as the peripheral protein. So let's label it, that is a peripheral protein. Those are the integral proteins and other thing, carbohydrate, cholesterol, all these things are there. I am going to explain you active transport and passive transport on the basis of this diagram. If outside the cell, directly coming to the point, if the outside, this is the outside of cell and this is the inside of cell. Outside the cell, very high concentration of oxygen is there, and inner side very low concentration of oxygen is there. Then oxygen is travel from higher concentration to lower concentration. It means it is going to diffuse in this membrane and easily pass, because it is a gas, it is the smallest molecule. So it is diffused from this membrane and transport from higher concentration to lower concentration. This is known as diffusion. This is known as diffusion. Now, after that, here is glucose molecule, C6H12O6, and this is also C6H12O6. High high concentration outside the membrane, low concentration inside the membrane. But now it can't be diffused from this membrane because glucose is hydrophilic in nature, and most part of this membrane is made up from a hydrophobic lipid molecule. So C6H12O6 is not going to easily diffuse from this membrane. So for this integral protein help to diffuse out either from higher concentration to lower concentration. This is known as diffusion, but it is facilitated by one protein. It is a diffusion, but it is facilitated by one protein. That's why it is called as facilitated diffusion. So I am writing here, it is known as facilitated diffusion. In both cases, guys, molecule travel from higher concentration to lower concentration. In this, only membrane is used. In this, protein is responsible for facilitated diffusion. But in both cases, there is a high concentration and low concentration. So when there is anything transfer from high concentration to low concentration, it means it does not require ATP. And if ATP is not required, then it is known as active transport or passive transport. My question to you, if ATP is not required, then this transportation is known as active transport or passive transport. I am waiting in the comment section. 22 student in the comment, 22 student in the chat. Everyone, I'm waiting for your answer. Kushi Naidu, thank you so much. Gurkirat, Taksh, Mayank, Gargi, Lalita, Vaishnavi, Mayank. Okay, all my students are joined in this chat section. I'm waiting for your answer, guys. I'm waiting for your answer. It is the active transport or passive transport? Facilitated diffusion, and diffusion is the active transport or passive transport? Arhan, thank you. It is passive transport. Very good. It is a passive transport. Because energy is not required. ATP is not required. If ATP is not required, then it is known as the passive transport. But if I am talking about the potassium ion, high concentration of potassium ion, for example, I am giving you a hypothetical example. If high concentration of potassium ion are present on the outside of the cell or membrane, and low concentration present inside. So naturally, there is a moment which is happen from higher concentration to lower concentration, from outside to inside. But now in that case, in that particular case, we required potassium outside the cell. Then for this low concentration to high concentration movement, we need energy. Without energy, we can't perform a active transport. It means a transportation of potassium from inside to outside with low concentration to high concentration, and this is known as active transport because it required ATP. Let's move to the another concept. Did you understood this topic, guys? Those are the points, and this is the diagram. I'm trying to elaborate all the point on the basis of this. On the basis of this concept, there is one question in front of you. They are asking, match the column 1 and 2 and choose the correct combination. Here in a column 1, neutral solute, water, polar molecule. Column 2, active transport, passive transport, and osmosis. Go for it. Go for it. Go for it, guys. Yes. The question is very simple. Question is very simple. Thank you so much, Taksh. Taksh is saying east or west. Mukul sir is best. Okay, thank you. Thank you so much, Tux. Not only in east, west, north and south also. B is the correct answer. Okay, let's check. Water. Water always travel from higher concentration to lower concentration through semi-permeable membrane, and it is known as osmosis. So the B3, B3 is in only one answer, B. Let's check other option, A2, neutral solute. Yes, passive transport and polar molecule, active transport. B is the correct answer. One another question, if we have the question in front of you, it is related to the endomembrane system, and you know that endomembrane system have endoplasmic reticulum, Golgi apparatus, lysosome and vacuole. This is a match the column. Match the column. Everyone try to answer. Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi complex, lysosome, and those are the explanations. Anyone? Anyone? Very good, Mayank, Sameer. Very good. Spiritual light. Okay, right answer. Unique name on YouTube. Unique captions or Instagram. Spiritual light. OP Arhan. Yes, spiritual light is saying answer is A. Let's check spiritual light is right or wrong. Match column 1 with the column 2 and select the correct option. Rough endoplasmic reticulum. You know that rough endoplasmic reticulum are responsible for synthesis of protein. Any option, protein synthesis? Yes. A3. Second, smooth endoplasmic reticulum is responsible for lipid synthesis. Any option? Yes. B2. Golgi complex, it's move material out of the cell or it have a digestive enzyme? No, it move material from out of the cell. And last one is the lysosome. It is a digestion required for intracellular and extracellular digestion. So A3, any option A3, only one option is there. A is the correct answer. B2, yes, right. C4 and D1. Let's move towards the another bank question. Assertion and reason question are very important for NEET examination. There are tricky question of assertion and reason, and every time you suffer in this type of question. So assertion is front of you. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome and vacuoles. And second one is the reason. Mitochondria, chloroplast and peroxisome are not the part of endomembrane system because their function are not coordinated with the same. Assertion is also in front of you. Reason is also in front of you. Check it out. Which option is correct according to you? Let's check. Let's check. Look, the endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, vacuoles. It is a correct statement. Second, mitochondria, chloroplast, peroxisome are not interconnected with each other. They have their what we can say individual existence in the cell. So this is also correct statement, but it is not exact reason why it is connected and it is interconnected. It is not an exact reason. So assertion and reason is true, but reason is not a correct explanation of assertion. So your answer is, if both assertion and reason are true, but reason is not correct explanation of the assertion, B is the correct answer. Let's move to the next important topic, that is the cilia and flagella, and it is most difficult topic of this chapter because mitochondria you studied in the class 9, 10th. Chloroplast you studied in the class of 8. Mitochondria is known as the powerhouse of cell. Chloroplast is the kitchen factory. All those things you know. But cilia and flagella in detail you didn't understood before class 11th. So, and this is one of the difficult topic also. Look, I am reading the statement. If you are getting the point, then it's okay. If not getting, I am going to explain this. Their core axoneme have many microtubule running parallel to long axis. Second point, axoneme have 9 pair of doublet, peripheral microtubule 9+2 array, central sheath, radial spoke. Is that the familiar topic for you? No. It is not a familiar topic for you. So let's draw the diagram on the basis of point here and try to understand this concept, that is the cilia and flagella. So first point, in very first point, they say that their core, that is the axoneme, has many microtubule running parallel to the long axis. Means they are having many microtubule. I am drawing here a microtubule, one, and second, third, fourth, fifth, sixth, seventh, eighth, ninth. Those are the microtubule running parallel to each other. And if we cut this section and observe, for example, this is the running microtubule. I am cutting the transverse section. If we cut the transverse section, if we get the transverse section, it means you are getting this circular appearance. So the circular appearance I am drawing here with the help of this. For example, this is a circle, and just going to mention a 9 point, because they are saying axoneme have a 9 pair of doublet. That's why 1, 2, 3, 4, 5, 6, 7, 8, 9 doublet. And this diagram I am going to erase because I draw this 9 doublet. So doublet I am drawing, one inside, one outside. One inside, second one is the outside, inside and outside. 9 peripheral doublet I am going to draw here. Let's assume inner one is the A and outer one is the B. Now those are the 9 peripheral doublet, and 2 central, they are saying that, and a pair of central microtubule. That's why this is known as the 9 plus 2 array. Now central microtubule I am drawing here, 1 and 2. This is completely known as 9 plus 2 arrangement. If they are going to ask you the question, what are the arrangement of cilia and flagella, you have to answer 9 plus 2 array. If they are asking you about the centriole or centrosome, only 9 plus 0. There is no central microtubule in case of centriole and centrosome, but in case of cilia flagella there are 2 central microtubule. Thus 2 central microtubule. Just read the point. Just try to read the point and understand the concept. The central tubule are connected by the breech and enclosed by the central sheath. So this is connected by breech. This is the interdoublet breech, and it is having one sheath, that is known as the central sheath. So this part is central sheath, this is the central microtubule, this is the breech, and this is the peripheral doublet. Now the sheath is connected to the peripheral doublet by using the radial spoke. So I am drawing here a radial spoke from this one of the doublet. Those are the radial spoke. So my question is to you, in this diagram, how many radial spokes are present? My simple question is to you, in this diagram, how many radial spokes are there? How many radial spokes? I am waiting in the chat section. Bhushan sir, thank you so much for your comment. Samir. Yes, Samir is saying, spiritual light is saying 9, right? If there are 9 peripheral doublet and each peripheral doublet is connected with the central sheath, it means there are 9 radial spokes. So they are saying 9 radial spokes. And the peripheral doublet are also interconnected by linker. It means the peripheral doublet also interconnected. So for example, this is a A, this is a B. This is a A, this is a B. A is connected with B with this linker. 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 This is a complete diagram of cilia and flagella. And the movement of this central sheath or central tubule takes place like this. Due to this complete structure move, and that's why this the movement of cilia and flagella takes place in the prokaryotic cell or eukaryotic cell also for locomotion. Now, this is a diagram, peripheral microtubule. This is from NCERT. Peripheral microtubule, that is the plasma membrane. This one is the plasma membrane because this complete structure is covered with the help of plasma membrane. So this complete structure having one more covering, that is the plasma membrane. Now, then central sheath, central microtubule, this is the bridge, and this is the radial spoke. So on the basis of that, I am having question for you. This is the diagram. If it is not clearly visible, I am writing here A, E, D, F, C and B. Let's try to answer. This is A. This is B, C. On the basis of that, this is D. This is F. Let's try to answer. This is E. D, spiritual light, answer is D, and D, I think it is a correct answer. A is a plasma membrane. Any A is plasma membrane. Yes, A is plasma membrane. Here also, here also. Now, second, B is a linker. B is a linker. B. Interdoublet linker or interdoublet bridge. C is, we can say that radial spoke. C is a radial spoke. C is a radial spoke, right? D is a central sheath. D is a central sheath. Yes, D is a central sheath. E is the peripheral microtubule. Yes, E is a peripheral microtubule. And F is a central microtubule. F is a central microtubule. D is the correct answer. Very good. D is correct answer. Next question in front of you. This is on the basis of chromosome. You know that metacentric chromosome, submetacentric chromosome, acrocentric chromosome, telocentric chromosome. So metacentric chromosome means a centromere at middle. So first point saying that centromere is present in the middle of the chromosome. So it is a metacentric. So this is the metacentric one. Two options are there, A and B. There is confusion. Now submetacentric. Second point, chromosome has a terminal telomere. If centromere at the terminal position, it is known as the telocentric. It means 2 is belonging to the telocentric. Telocentric 2, this is the correct answer. Just read the statement and try to answer. And if you want this PDF, just comment PDF in the comment section. So I'm going to provide you this PDF. Now, next, here there are 42 minutes completed and one chapter revision is completely over. That is a cell, the unit of life. Now we have 18 minutes, and in this 18 minutes, we are going to cover a cell cycle and cell division. If any question on the basis of cell, the unit of life, you can ask in the comment section. If you are not live, if you are watching this recorded as a recorded session, you can comment a question on my comment section. I am going to answer you these questions. Okay, so if any question, let's ask in the comment section, or we are going to move further. Suraj saying PDF, sir. Okay, PDF, definitely. Give me a 1 minute of time, guys. In 1 minute, I am going to start a next topic, cell cycle and cell division, and very important topic in which I am going to cover a cell cycle as well as very important phase, that is the meiosis 1, prophase 1, lactotane, zygotane, pachytane, diplotane and diakinesis. Important part of the cell cycle and cell division. Give me a 1 minute time only. let's start with the next topic, cell cycle and cell division. In very first point, they are explaining us that duration of cell cycle varies in each organism and each type of cell. Giving us the example, duration of eukaryotic cell, that is human cell, it takes a 24 hour to divide. In yeast cell, 90 minutes. In bacterial cell, 20 minutes. So it varies cell to cell. But this complete cell cycle means their growth, their division, their development from one daughter cell to the two daughter cell, and their development is known as the cell cycle. And what is cycle in general? You are studied TCA cycle, you already studied a phosphorus cycle, you already studied the carbon dioxide cycle. What is cycle? Cycle means anything start from one point and at the end it comes on the same point. Cycle is like a karma. It start here and end here. If you are doing any good thing in your life, definitely it will be reflect in your future life. And any bad thing you are doing in your life, then definitely it hits you in upcoming future. So do good thing in your life. Always try to do best for everyone. Okay, let's start with the cell cycle. Interphase, M phase. Interphase is the first step of the cell cycle and M phase is the second step. Interphase is also known as resting phase because division is not active. But in this phase, metabolically cell is very active because it is preparing himself for cell division. First point, it is a phase between two successive M phase. It is phase between two successive M phase and it includes cell growth, DNA synthesis, everything. It lasts more than 95% of the duration of cell cycle. It means 95% time cover in the interphase, 5% time covered into the M phase. If we are talking about the 90-minute time, it means 82 minutes is for only interphase, 8 minutes for M phase. If we are talking about the 24 hour, 23 hour for interphase and 1 hour for M phase. Now, this is a handwritten notes. I'm coming with these handwritten notes for you, and this complete cell cycle I draw here. First one is a M phase. Look at the M phase. From this point to this point, M phase. M phase include prophase, metaphase, anaphase, telophase. And if I am talking about prophase, metaphase, anaphase, telophase, it comes under the karyokinesis. That is the nuclear division. And after that, successively, simultaneously, one division takes place that is known as cytokinesis. So M phase is divided into karyokinesis and cytokinesis. So if understood this point, if there is one cell is diploid cell, 2n number cell, and the cell first of all enter into the G1 phase, that is the interphase having three stages, G1, S and G2, G1 phase. What is going to happen with this cell in the G1 phase? G1 phase, in G1 phase, continue growth of cell takes place. Continuous growth, size of the cell going to be increases. Increase in the cell organelles, mitochondria, chloroplast, those are cell organelles which is going to increase in the G1 phase. RNA and protein synthesis. Now, protein synthesis, which kind of protein is synthesized into the G1 phase? So you know that after G1 phase, which phase is going to come? S phase. S phase means a synthesis phase. In this synthesis phase, DNA replication takes place. So in G1 phase, the proteins are synthesized which is helpful for S phase, like helicase, like DNA polymerase, like ligase. Those are the single stranded binding protein. Yes, those are the proteins. Those are the enzyme, but all enzymes are protein. So those are the enzyme or protein which is synthesized into the G1 phase for S phase. Now, after that, S phase is there, interphase, long time, 95%. So S phase is a synthesized phase, that is the replication of DNA takes place. That is a nuclear DNA replication. Understood the point? Cytoplasmic DNA replicate at any stage. At any stage, but the nuclear DNA replicate only in S stage. Now, synthesis of histone protein which is responsible for packaging of DNA is also synthesized in case of S phase. DNA 2C means this is a C is a DNA content value. So in this phase, DNA duplicate, DNA doubles. So 2C if there is a content, after S phase is become 4C. But chromosome number is not going to double. If chromosome number is 2n, then it is a 2n after that S phase. Very important. But sometime a tricky question asked by NEET. Chromosome duplication. Number of chromosome doubling is different kind of concept. If they are saying chromosome duplication, yes, chromosome duplication is takes place. They are talking according to the DNA content. According to the DNA content. So chromosome duplication takes place. Chromosome number doubling not going to happen. DNA content double, yes, it is takes place. And centriole duplication, yes, it is takes place in case of S phase. Now G2 phase. G2 phase, tubulin protein synthesis, cell grow continuously, actual preparation of N phase. Very important point, tubulin protein. Tubulin protein is responsible for formation of spindle fiber. Spindle fiber we required in which phase? M phase. That's why G2 phase is responsible for formation of tubulin protein. So this is another part of cell cycle. I am giving you just briefing of the checkpoint. There are 3 checkpoints. One checkpoint is present here, second checkpoint is present here, and third checkpoint is present here. This checkpoint is known as G1S checkpoint, this checkpoint is known as G2M checkpoint, and this checkpoint is known as spindle checkpoint. Those are the checkpoint which is responsible for checking of genetic material, because genetic material is very crucial. On the basis of genetic material only our appearance, our brain capacity, our retardation, our height, everything is depend on the genetic material. So this checking is very important during the cell division. Let's directly come, because we have only 9 minutes with us, 9 not 8 minutes only with us. So in that 8 minutes, I am going to explain you the meiosis. In meiosis, meiosis 1, in meiosis 1, prophase 1, in prophase 1, 5 phases. How many phases? 5. 1, lactotane. Second, zygotane. Third, pachytane. Fourth one is the diplotane, and fifth one is the diakinesis. Let's start with the leptotane. First of all, in leptotane, small breakdown of the nuclear membrane start, because it is a prophase. Not completely disappear, but small breakdown of nuclear membrane, and all the chromosomes or chromatide or chromatin fiber present into the nucleus attached to one end of nuclear membrane, and it is looking like a bouquet. It is looking like a bouquet. That's why lactotane sometimes known as bouquet stage. Second point, zygotane. Thus condensation start here. So condensation start like this. Now this condensation become more closer. This condensation become more close, and ultimately it look like this. So this is a phase in between lactotane to zygotane, and after lactotane there is zygotane. In zygotane we are directly observed two homologous chromosome pairing with each other. Now homologous chromosome come close to each other. Homologous chromosome come close to each other is known as the synapsis. So where synapsis takes place in case of zygotane, this is known as the dyad or bivalent. This is known as a dyad or bivalent. Thus chromosome, homologous chromosome come close to each other, but it can be far apart from them, each other. So the sticking or a joining of chromosome we need until a diplotane. So for this purpose, there is one complex responsible for that, that known as the synaptonemal complex. Thus two homologous chromosomes join together with one proteinaceous complex, that is known as the synaptonemal complex. After that, this dyad is converted into the tetrad. 4 arms are there in tetrad. Thus 2 are a sister chromatide, and thus 2 are the non-sister chromatide. Non-sister chromatide of homologous chromosome. Homologous chromosome means thus come close, and non-sister chromatide, which is not a same chromatide. Non-sister chromatide of homologous chromosome just start a crossing over. It start crossing over in zygotane only. But look, is it crossing over? Yes, according to you, it is. Yes, it is start in zygotane. But is that a crossing over? No, it is not stick with each other. So this kind of image are formed in case of zygotane. but it is far away from each other. This complete joining takes place in the pachytene or pachytene. Crossing over takes place, non-sister chromatide of homologous chromosome or genetic material is going to be exchanged. Recombinase enzyme is responsible for that, and recombination takes place in case of pachytene. This point have to remember. You have to remember this point. Thus 3 phases we already discussed. That's why I take it as a reference. Lactotane, zygotane, pachytane. Now diploatane. In diploatane, chiasmata formation takes place. Chiasmata, this thing, this crossing over looking like that. But synaptonemal complex breaks. These 2 homologous chromosomes far away from each other. It means a synaptonemal complex is break, and it is not break like this. It start breaking like this. Like that way it breaks. So synaptonemal complex break here, and this is known as the separation of homologous chromosome. This is also known as disjunctioning of the homologous chromosome. And lastly, in diakinesis, all the changes occurs in prophase. That is the what we can say, disappearance of the nuclear membrane, disappearance of the nucleolus, formation of spindle fiber, movement of the centriole to the opposite pole. All those things carry forward or happen in case of diakinesis. Aster ray formation, all those things is happening in case of diakinesis. So in this slide also I have explained lactotane, zygotane, pachytane, diploatane and diakinesis. On the basis of that, I have some question for you guys. First question in front of you, a cell examined during prophase. The question is, the cell examined during prophase of a cell cycle contain 100 unit of DNA and 50 unit of chromosome. What would be the number of DNA and chromosome in anaphase? You have chance to answer the question. Yes, Jadhav. Ragini Bhalme, S.A. Genius brother. You are answering the question, good. 100 unit of DNA and 50 chromosome. What will be the chromosome in the anaphase? Anaphase, you know that in anaphase, there is a centromere and there is an attachment of spindle fiber. So from one chromosome, 2 chromosome formation takes place. So chromosome become double. Means 50 chromosome becomes 100, but genetic material is divided same. So DNA content is only 100 picogram, 100 units. So 100 picogram DNA and 50 to 100 chromosome. It means answer is 100 DNA and 100 unit of DNA and 100 number of chromosome. 100, yes. C is the correct answer. Next question in front of you. Which of the following is correctly matched? Lactotane, diploatane, pachytane, zygotane. Lactotane for formation of bivalent, diploatane chiasmata appear, pachytane chiasmata terminalization, and zygotane formation of bouquet. You know that bouquet is formed in which case? Bouquet is formed in case of leptotane. And they are giving in a zygotane. This is incorrect, and they are asked correct. Pachytane, chiasmata terminalization, it in case of diakinesis, not case of pachytane. So this is also wrong. Chiasmata appear, diploatane, yes, it is correct. It start appearing in case of pachytane and completely appear in case of diploatane. B is the correct answer. Next question. A diploid cell before S phase, quantity of DNA is 20 picogram. Before S phase. Now in S phase is duplicate, you know that. Before S phase is 20 picogram Pg. Okay? And this is S phase. After S phase it becomes 40 picogram. Okay. Let's read the question. After meiosis 1, okay, they are not asking after yes. They are asking after meiosis 1, what will be the quantity of DNA in each daughter cell? After meiosis 1, chromosome also divide. Na? For example, 2n number cell is there after meiosis 1. After meiosis 1, this 2n number cell is divided into n and n. So, if 20 picogram, after S phase become 40 picogram in 2n number cell, and this 2n number cell is divided into a nn number cell. So 20 picogram in one and 20 picogram in other. So 20 picogram is correct answer. Any option having 20 picogram? Yes, B number option having 20 picogram. Next question, and I think so last question of today's session. Number of chromosome pair are at equator in the metaphase of the diploid plant, n is equal to 25 shall be. Now number of chromosome is 50, but they are asking about the pair. So 25 chromosome, pair also 25. 25 is the correct answer. I hope so you understood complete topic of the cell, the unit of life, and cell cycle, cell division. One more time, thank you for joining everyone. Thank you for your answer, your response in the comment section. And upcoming days, we are going to discuss anatomy, morphology, and all this chapter of the botany with you. If you want this PDF, if you want this complete PDF, then just comment section, in comment section, type your mail ID or write it down, PDF. I am going to send you the PDF. Thank you so much for joining once again. Bye-bye and take care.